Like he said, my name is Kelli Jestes. I'm a second-year BMS student here working in his lab. So in Lowry's lab, the focus is on osteoporosis, and my project more specifically is trying to elucidate the molecular signatures that's associated with elevated bone formation rate. So as you guys have been learning, bone is a very living, dynamic tissue, and its integrity and its strength relies on osteoclast breaking down old bone and osteoblasts forming the new bone or putting in the new bone. And according to the National Institute on Aging, sometime around age 30 is when we hit our peak bone mass. And as we get older, the bone is breaking down more than it is replaced, and it puts individuals at a higher risk of osteoporosis. So what exactly is osteoporosis? Osteoporosis is a silent disease of decreased bone density due to osteoblasts working faster or harder than osteoblasts, so that resorption is exceeding the bone formation. It causes bone to become super brittle, and so when you look at this picture down here, you see that there's extremely less density in this picture versus the normal bone mass here. And so individuals with osteoporosis have a higher risk for fracture and disability. I mentioned it's a silent disease because a lot of the times it's not noticed or diagnosed until the bone actually breaks. And so individuals can develop this over a really long period of time. And then women are especially at risk if they have family history, if they smoke, or if they're postmenopausal. So why do we care about this? Well, osteoporosis is common, it's costly, and it's serious. About 10 million Americans have osteoporosis and 44 million have low density. 1 in 2 women and 1 in 4 men will break a bone due to osteoporosis. A woman's risk for fracture is equal to her combined risk for breast, uterine, and ovarian cancer. In the United States, hospitalizations for osteoporosis fractures exceeds those for heart attack, stroke, and breast cancer, which is alarming. It's costly. Osteoporosis-related bone breaks cost patients, their family, and the healthcare system about $19 billion annually. And experts predict by 2025 that osteoporosis will be responsible for 3 million fractures, resulting in $25.3 billion in costs. And what's really sad is that 25% of hip fracture patients will die within a year of injury. And every year, of the nearly 300,000 hip fracture patients, a quarter of them will end up in nursing homes, a half will never regain their function. So essentially these patients are losing their independence and their quality of life. The only current therapies are anti-resorptives, and there's only 2 FDA-approved anabolic treatments, but they're extremely expensive, which I'm sure you guys have learned about. So this just states that it is so important for us how important it is that our research is going further and trying to find a new therapeutic for osteoporosis. So now that I've given you all the facts, let me move on to what we actually do in the lab. But first, I need to give you guys a little bit of background information. So BMP is bone morphogenetic protein. And we look at its signaling in bone because the BMP superfamily is a mediator of the skeletogenesis. The BMP pathway is associated with promoting bone formation, and the activin pathway, it promotes bone loss. But keep in mind that this is a generalized statement for osteoblast lineage. It's a classic paradoxical signaling where one side says to stop and the other side says to go, and the cell has to decide who to listen to pretty much. And Madeline will talk about how these 2 pathways compete. So I will let her do that. So I will just teach you guys essentially how these pathways work. They have 2 types of receptors, type 1 and type 2. Type 1 is this ALK236 for the BMP pathway and ALK47 for the activin pathway. And the type 2 receptors are BMPR2 and ACVR2A and B. And as you can see, that receptor is used for both the BMP and the activin pathway. So what happens is type 2 receptor dimerizes to the type 1 receptor. And then when the ligand binds, the type 2 receptor will phosphorylate this type 1 receptor. And then the type 1 receptor will phosphorylate this SMAD158 for the BMP pathway and the SMAD23 for the activin pathway. The mouse model that we work with in our lab knocks out this BMPR2 receptor. And when we knock that, or when we remove that BMPR2 receptor, you can see that it leaves a competition for this ACVR2A and B, which is the only type 2 receptor available for these ligands to bind and dimerize with that type 1 receptor. So this increases the competition for that shared pooled receptor. Okay, so let's stop and recap. We have data that's presented here that generally activin pathway is promoting bone loss and the BMP pathway is promoting bone formation. And our model knocks out this BMPR2 receptor. So what do you guys think the signaling would be when we're knocking out this BMPR2? Would SMAD158 be up, down, normal? And then what would SMAD23 be? Up, down, normal? Does anybody have any thoughts on if we're knocking this out, what would the signaling be? Yeah.
SMAD would be going down. SMAD 1.5 would be going down on the left, and then 2.3 would be upregulated.
Good. So that's what you would think we would see. However, surprisingly, our lab discovered that it was the opposite. That the competition for this ACVR2A led to the reduction in activin pathway signaling. And you can see that with this Western blot here. This BMPR2 flox is acting as the control, and this BMPR2 conditional knockout mouse is what we are working with. So you can see right here that the level of activated SMAD2/3 was substantially reduced, and there was no apparent alteration in this BMP pathway looking at the SMAD158. So that means that the activin pathway is being sequestered somehow when we knock out this BMPR2 receptor. So another question, if this activin signaling is reduced, what do you guys think the skeletal phenotype would be? Would we have high bone mass, low bone mass? What do you guys think? Speak up, can't hear you. You have it here in front, say it.
High bone mass.
High bone mass, hey, you were right, good job. Okay, so looking at this graph, you see that this black line is the control mouse, and then this red line is the BMPR2 mutant mouse. So when we knock out the BMP receptor 2, not only do these mice have the elevated bone mineral density, but it also shows that the age-related decline is reduced, which you can see that the mutant mice lost approximately half of what this control mouse did here. And this is just milligrams hydroxyapatite per cubic centimeter. So you're just seeing here that it lost less than the control did. So the question now is, how is this possible? Is it due to osteoclast activity decreasing, or is it due to osteoblast activity increasing? So they took serum from the mice and ran ELISAs to measure the activity of both these osteoclasts and osteoblasts. And CTX is an osteoclast marker. And as you can see that it was unchanged between this 35-week mark and the 55-week mark. However, when they looked at osteoblasts, so P1NP is an osteoblast marker, they found that at 35 weeks, this osteoblast activity was significantly higher at 35 than at 55. And then at 55, it actually went down to relatively normal. So this establishes that the high bone mass is associated to an increased osteoblast activity. Dr. Hum?
Yeah, so can you explain 35-week, 55-week, what that would represent in terms of like bone health? Like how old would you equate this to in, say, a patient?
So like I said, our peak bone mass is at around 30 years old. And so what you're seeing here is that the osteoblasts are increasing, but at the 55 week, they're going back down. So usually osteoblasts stop producing or stop working at a higher rate, or the osteoclasts are more in abundance and have the high activity, that it's decreasing that bone density. So with these mice, we see that at 35 weeks, they are increasing and then went down to relatively normal at 55 weeks, showing that these osteoblasts are a little bit higher than what we normally see in a normal model.
So would 55 be considered an old mouse?
Yes.
Okay.
Yes. Did you have a question, Dr. Larson?
I was just going to ask what the lifespan of a mouse is.
Oh, and honestly, I don't think I know that.
Not more than 2 years.
2 years? Okay. So yeah, mice do age faster than we do. So this is showing 55 week is an older patient in our case. So, okay. Let's see. So we then took the bones from the BMPR2 conditional mice at 35 and the 55 week, and we're trying to figure out what exactly is going on that's driving this 35 weeks, or this osteoblast to be, have a higher activity at 35 and at 55. And we think that there is some kind of alteration in this cell's communication and gene expression that's associated with this elevated bone formation rate in BMPR2 knockout mice. And our approach is to look at the protein signaling and then work our way down to the gene signatures and then use candidate-driven approaches to hopefully find the needle in the haystack. So let me just stop right here, and before I explain this, I just wanted to stop here and give you guys a little bit of a tip. If you are thinking about doing the research thesis track, I highly recommend you guys look for opportunities where you can get on more than one publication. So for example, these antibody arrays are a brand new technique, and because I've established it in our lab, it has allowed me to collaborate with other labs that are doing similar things, and I can use this technique for their project to get on a paper with them. So I just wanted to kind of throw that plug in that if you are thinking about doing this, it's a great opportunity to get your name on multiple publications than just the one. So I highly encourage you guys to use those opportunities. Okay, but anyways, let's get back to science. So we took advantage of a new technique that allows us to simultaneously detect about 1,300 targets using just a small amount of protein. And for those of you that have ever done Western blots, this is huge because with Western blots, you can only look at just a few of the targets using a lot more protein. So this is called a high throughput phosphoprofiling antibody array. In the simple terms is that we are looking at what proteins are being phosphorylated and exactly where on the protein are those being phosphorylated. So it works. So how it works is that we take a protein lysate from our mutant mice, and we then add biotin to it, and that labels the protein. And then we add the protein to these glass slides that have antibodies embedded on them and are against each target. Then we add a fluorescent tag right here. We send them off to a company called Full Moon Biosystems, and then we essentially get two things back. We get a pretty slide that you see here, a picture of this, and each one of those bright green dots represents a protein that has bound to an antibody. Then we also get back an Excel spreadsheet showing the quantitative data. So when we get the Excel spreadsheet back, we try to normalize these against housekeeping genes, and remember that the osteoclast activity, again, was the highest at 35 weeks and went back to relatively normal at 55 weeks. So we wanted to find those proteins that had a fold change less than or equal to 0.75, or in other words, that the phosphorylation was reduced, and at and that were relatively normal at 55 weeks. And those proteins that had a fold change of greater or equal to 1.5, or the phosphorylation was increased at 35 weeks and relatively normal at 55 weeks. So I know this is really hard to read, but this is these are the data that show those proteins that had reduced phosphorylation at 35 weeks right here, and that were relatively normal at 55 weeks. So essentially, we have about 40 proteins that are repressors of the osteoblast activity. Yeah.
How do you determine 0.75 and 1.5?
So we didn't want to go too low and then really, get rid of a lot of proteins. So we kind of found this was a sweet spot to give us just enough proteins that we can really look into. So it was kind of under our discretion. Any other questions? Okay, so in these are the proteins that at 35 weeks where the phosphorylation was increased, and then again at 55 weeks, they were relatively normal. So with both of these lists, we have about 86 proteins relating to this elevated bone formation rate for these BMPR2 mutant mice. So what's next? At this point, we don't know what these data mean, and we're literally trying to find the needle in the haystack. But we're going to do bioinformatic analyses to start putting these linear pathways and then evaluate if any of these will inform us of the gene signature that are associated with this. So in order to find a gene signature, we previously performed genome-wide transcriptome profiling in bones at 35 and 55-week-old control and BMPR2 mutant mice. Any gene that displayed a twofold or greater expression compared to the housekeeping genes was marked as upregulated, and any gene with a twofold or lower expression level were considered downregulated. The genes were sorted and were marked as either 35 or 55 weeks because, again, the osteoblast activity was higher at 35 weeks and relatively normal at 55 weeks. And we found that there were 120 genes that were upregulated at the 35-week mark, and there were 131 genes downregulated at the 35-week mark, which is great, but this is still a lot of genes to go through. So we wanted to do the RNA-seq again with the 55-week-old mice. And we found that 2,381 genes were upregulated at 55 weeks, and 172 genes were downregulated at 55 weeks. And then there's a table that was constructed showing comparisons between these genes that were up and down regulated at the 35 and 55 week, because we currently don't know if whatever is driving the osteoblast activity is an overexpressed driver or an underexpressed repressor. So we had to consider all of these a possible option. So after further investigation and extensive lit searches, we found that only 9 transcripts have been previously implicated in bone physiology, and we consider those the genes of interest. And here they are. So out of the 9, we have 5 putative repressors, which are NMU, B2M, FGF1, SMPD3, and phospho1. We have 3 putative drivers, PAC4, RPL38, and NDFIP1. And then we have 1 hybrid, which is inhibin beta E. So we performed the RNA-seq using pooled samples. So we wanted to validate these individuals using candidate-driven qRT-PCR. And at 35 weeks, we validated that PAC4, RPL38, and NDFIP1 were indeed up at 35 weeks. This BGLAP is an osteocalcin, which is just for a control, and that was normal at 35 weeks, which is saying the exact same thing that the RNA data or the RNA-seq data determined. And then at 55 weeks, PAC4, RPL38, and NDFIP1 were all relatively normal. And again, this was normal as well. And that was saying the exact same thing that the RNA-seq data had showed. So for a summary, we have a BMPR2 mutant mice that displays high bone mass. We have gone through antibody arrays, antibody signaling arrays that identified 86 proteins that can act as either a repressor or a driver of osteoblast activity. We did genome-wide transcriptome profiling and found 179 differently expressed genes associated with increased osteoblast activity. And then several of those genes actually correspond with osteoblast differentiation and activity and are upregulated in these mutant mice. So collectively, our findings provide insight into the mechanisms regulating age-related bone loss and highlight potential targets for therapeutic for bone mass.